Good morning, everyone, and welcome to the Precision Health Virtual Symposium. My name is Vicki Ellingrad. I'm the Associate Dean for Research in the College of Pharmacy, as well as the Education Lead for Precision Health. We're very glad that you've joined us here today, and know that you are not alone because we have almost 500 people that have registered for our symposium. We're very excited about that, especially since the people that have registered are not only our standard researchers at Michigan, our research staff, but also include our students, our trainees, our postdocs, our industry partners, clinicians, colleagues from other research institutions, and most importantly, our interested community members. So we warmly welcome everyone who is joining us today. Our agenda for the morning includes guest speakers and researchers, followed by a poster presentation or a poster session, and then in the afternoon, talking about just what Sachin brought up is how we're going to be using, how we have used the data already that so many people have entrusted to us. We have a few scheduled breaks throughout the day, so you don't have to miss anything to step away. And it's going to be my job to make sure that we stick to our agenda as much as we can. If you have any questions or problems with the webinar today or throughout the day, please feel free to contact Tina Krazier. I think she probably just put her email, nope. Her email is tcreguer@umich.edu, and she can also put that in the chat so that you would have that handy. If you have questions for our speakers, and I sure hope that you do, please use the Q&A tab on your Zoom screen. It's on the bottom. I will be monitoring that tab along with some of the Precision Health staff, and we'll be able to share questions with the speakers during the discussion periods that we have built in. The focus today of this event is the intersection of research and community, how participant data is gathered and used in research, and how that data is now being used to improve patient care. I'm very excited about this. Our morning session is going to focus more on research and some of the important lessons learned, because as we've learned, we don't want to repeat the mistakes of our past. And so we're really hoping that we can learn from each other to become a stronger precision health community. Midday, we'll have a poster session that will be featuring the work of researchers who have been funded through Precision Health. And then in the afternoon, we'll be coming back to focus on how precision health data impacts health and patient care. With that, I want to introduce our morning keynote speaker. Dr. Nancy Cox is the Mary Phillips Edmonds Gray Professor of Genetics, Director of Vanderbilt's Genetics Institute, as well as Director of the Division of Genetic Medicine. She is a quantitative human geneticist with a long-standing research program to understand how genetics affect common human diseases. We are honored to have Dr. Cox serve as a member of our National Advisory Board because her insight and guidance on how matters relating to engaging communities for research is so vitally important, and actually the springboard for why we're doing the symposium today. Her own work at Vanderbilt involves working with an initiative called BioView, which has data from more than 240,000 subjects. She has earned multiple prestigious awards for her landmark research, and we are so happy that she is here with us today. With that, Dr. Cox, I would like to warmly welcome you to present your research on how BioView has used community engagement in its development and growth.
Right. Thank you. It's a pleasure to be here. Let's see. Have I successfully gotten my slides up?
Yes, you do.
All right. Thank you very much for the opportunity to be here. It's a real pleasure, and I have been looking forward to this. I always enjoy visiting at the University of Michigan, and so I wish I were there. So topics for today, I'm going to describe some of the basics of our biobank and the involvement of the community board from the beginning. I'm going to give some examples of the unique research that I think biobanks enable, and really just vignettes of many different projects that are underway in the biobank today. And at the end, I'll give a bit of a case study of the most recent interactions with the community board around some expansion of BioView and the ways that our community of scientists interacts with the community board in the context of the biobank. So everybody appreciates what a biobank is. We have subjects who agree to give up some parts, usually just blood for DNA or other samples, but sometimes in the context of surgical medical care, sometimes additional parts of the body. And there must be some information on the phenotype. That's sometimes by self-report. It's sometimes by electronic health record. Sometimes people undergo a battery of tests. It depends on the kind of biobank. And then we often have some kinds of omics interrogation on those materials that is linked to the health information that people provided. And that can come from the genome variation, but it can come from the measurement of other omics now as well, measurement of transcriptomes, measurement of metabolomes, methylomes, all the omics. Our biobank is called BioView for Biobank at Vanderbilt University. We now have about 285,000 specimens in the biobanks of DNA samples. Our biobank is linked to electronic health records, and that is really the only way that we have phenotypic information on our patients at this point, although there are pilots for more wearable devices. Vanderbilt built their own electronic health records more than 30 years ago now. And so we have up to 30 years of electronic health records on some of these subjects. But on average, we have 10 to 15 years of electronic health records. We have genome variation measured in 120,000 people for genotyping array level data, genome-wide genotyping arrays. We have an additional 20,000 or so subjects with exome sequencing or whole genome sequencing, and we hope to bring that number up considerably in the near future. And then we have imputed transcriptomes and now are imputing metabolomes into these data. And I think the opportunity to do what we call imputation of omics also offers some really useful advantages, and I'll talk about that as well. So Vanderbilt's biobank was an opt-out at the beginning, and early concerns of the Community Advisory Board revolved around how to maintain the privacy of subjects, and in particular, questions were raised about who would know about whether someone was in the biobank or not. And so there was a lot of effort put into thinking about how to ensure that no one could know for sure if they were in the biobank, even if they hadn't opted out. And so as a consequence of this concern, eventually they came up with a strategy of randomly removing 10 to 15% of the subjects who were otherwise going to have been participants. So Vanderbilt extracts DNA from leftover clinical blood samples drawn for clinical purposes at essentially the moment that they would be thrown out 3 days or so after having been collected and all tests run. The DNA samples were sent to the laboratory for DNA extraction. And so they had an algorithm that would randomly remove 10 to 15% of the samples. This ensured that no single patient, no physician no BioView user could be sure who was in or out, even if the patient told the physician, well, I didn't opt out, and the physician knew the patient had had a clinical blood draw, the physician couldn't know whether or not the patient was actually in the biobank or not. And another interesting concern, really from our community board, was that although they were educated about the biobank and in full support of moving forward, there were concerns that some members of the community would push back after a rollout. And so to avoid that kind of failure to have educated as much of the community as necessary, they actually publicized the rollout, but didn't actually roll it out. So they had all the signs up in the hospital about how to opt out of the program. They publicized that this would be happening, but they didn't actually roll it out. And when there was actually no outcry, no pushback, no stated concerns from the community, quietly rolled out the program later on. But I wanted to raise these as just examples of the kinds of interactions that the scientific review community and the community advisory board have to try to work through the concerns raised by the community advisory board. Vanderbilt has gone to a prospectively consented biobank now. So in 2014, they went to a prospectively consented activity. But I think, so we all, the civic community, hoped they would up randomly removing subjects, but they considered it to be so much a part of the system that they've actually left it in. So we still randomly remove 10 to 15% of subjects that might otherwise be part of the biobank. That's really not such a hardship. We generally have a budget that reflects how many samples can be recruited over a given period of time. And so we recruit that many. It's just, for any given batch of samples of blood, 10 to 15% are removed randomly from the set. And I do want to dwell for a moment on what I think the really unique aspects of biobanking research reflect. So biobanks are much more akin in many ways to a cohort design, a cohort study, since you're really looking at, although it's a cross-section of individuals, you're going to be following many of them over time. And our special recipe in Tennessee, I think the things that you can do uniquely well in a biobank because of the breadth of the phenotype is to put genes and variants in and see what phenome comes out. Learning really what medical phenome is associated to genetic variants, to the genetically predicted gene expression, to polygenic risk scores for a given phenotype, all provide really interesting and important information that you really can't get in any other way. And so while you can do GWAS, genome-wide association studies, in the context of the genetic data in a biobank, and all biobanks certainly do those studies, you remember that the proportion of affected individuals for any given disease are merely going to reflect the rate in the population. And so when we want to study, as we do recently, COVID-19, our ability to ascertain patients is just dependent on what proportion of the population is infected and comes to our medical center. And Fortunately for Nashville, that's actually been a quite small proportion of subjects to date. So, but we have the historical data, and we can do things like go back and look at the risk factors for being hospitalized for pneumonia after a serious viral infection. And we find that the same predictors that we know are predictive of the worst outcomes with a COVID-19 infection are also seen with patients hospitalized for pneumonia after severe flu infections. And so there's a lot of historical data that we can bring to the question, even when our individual sample sizes for a disease are smaller. We do think there's a lot of value in imputing, so this is the example for gene expression, but we're imputing at scale metabolomes into the biobank as well. And the idea here is simply to take a reference panel of measured transcriptomes and genome variation, and we can use the GTEx data for this. The last of the GTEx papers have just recently come out. GTEx involved a collection of DNA with whole genome sequencing, as well as RNA-seq to measure transcriptomes in 49 tissues. And so we can build predictive equations from the genotype data for the transcriptome to measure what we call genetically regulated expression of the genes, so the genetically determined part of gene expression. What we often want to study are clinical traits and diseases, and so we'll think of that as the center of this circle, but a ring around that is what we might measure as gene expression from all different human tissues. Some part of what we measure as gene expression is genetically determined. But we know that many other factors affect what we measure as gene expression. And those include exposures, like whether you drank coffee this morning or some other caffeinated beverage. And if we were to take samples from the participants in this symposium today, samples from whole blood, samples from liver, samples from, and measure transcriptomes, we would be able to detect a signature of caffeine in those who indeed had a caffeinated drink this morning for breakfast. That was only partly dependent on the genetics and highly dependent on the exposure to the caffeinated drink very recently. So that the exact pattern might be partly influenced by the genetically regulated expression of genes, but the majority of the signature would be attributable to the recent intake of caffeine. So, and of course there are many factors, diet, lifestyle factors that affect the expression of genes in important ways. And we know that some part of why people develop disease over time, why they get high LDL cholesterol or high triglycerides, or eventually develop cardiovascular disease, can in part be attributed to the genetically regulated expression of genes, also to the changes in proteins that can occur through genetics. And we also understand well that there are many measurable exposures that affect those risks, as was mentioned this morning, including social determinants of health. So all of these factors influence our risk of developing a clinical trait, but once we have developed high LDL cholesterol, well, that feeds back on our measured gene expression. And so in addition to these exposures and the genetically regulated expression of genes, the traits that we develop over time, the diseases that we have, feed back on what we measure as gene expression. And for that reason, the directly measured part of gene expression has a fairly confusing pattern for understanding causality. We know that many thousands of genes can be differentially in a statistically significant way between cases and controls who have a disease, even when we measure accessible tissues like whole blood. But most of that is a consequence of this trait altered feedback rather than a cause of the disease. And so forgetting at the causality, this genetically regulated expression can be a good thing. And that's the main reason why we're interested in doing this imputation, not just for transcriptomes, but for other omics as well. These are very useful endophenotypes that are sort of occurring between the actual DNA and the development of disease. So phenotypes that help us understand how we get from the genetics to the disease. So we take these prediction models that were developed for the genetically regulated expression of genes and apply them to the genetic data in the biobank. So we haven't measured the transcriptomes of these subjects, but by calculating this genetically predicted expression, we can create kind of a gene by medical phenome catalog. So Hockey M and Eric Gamazon developed some of the first work on this. One of my postdocs, Dan Zhao, has a paper in Nature Genetics in Press with a much improved models for prediction, and doing the heavy lifting on the actual calculations are postdoc Tyne Miller and a young faculty member at Vanderbilt, Shui Zhang. And Anwar builds all of our portals where we see these results. And so you can query this gene by medical phenome catalog you can query it by gene, or you can query it by phenotype. And here's an example of the associations to a gene that's of some interest because it's been characterized as contributing to the risk of Alzheimer's disease, both rare variation, rare coding variation in heterozygous form increases the risk of late onset Alzheimer's disease, but also common regulatory variation that affects the expression of this gene has also already been reported to be associated with Alzheimer's and other dementias. Recessive mutations of this gene cause a Mendelian condition called Nasu-Hakola disease. And you can see that the phenotypes associated, the top phenotypes associated with genetically regulated expression of this gene do not include Alzheimer's. We do see cerebral laceration and contusion as a phenotype, but it's mostly about osteomyelitis. And we have hallucinations here. If we go further down into the nominally significant but not markedly significant phenome, we see many fractures, potentially consistent with the osteomyelitis diagnoses, and eventually start to see delirium, psychogenic disorder, dementia with cerebral degeneration, all nominally significant, but not nearly as significant as the osteomyelitis. The interesting thing about Nasu-Hakola disease is that it presents in the late teens or early 20s with painful bone cysts, usually due to osteomyelitis. And the bone cysts arise because bone's been resorbed without any bone being redeposited. And that the holes where the bone's been resorbed has a kind of fatty necrotic tissue that's a really good medium for bone infection. And so they get painful infections starting around joints, but then develop many fractures as the bone is eroded through the 20s, have this continual problem with osteomyelitis actually. And then by their 30s have a really severe early onset dementia, and most are dead in their mid-30s. So it's a really horrible disease. But seeing the osteomyelitis, the fractures, all make sense in the context of what's known of Nasu-Hakola disease. And and knowing the relationship to late onset Alzheimer's makes for an interesting pattern. That was really surprising when we look at the phenome associations of the G-Rex of presenilin, presenilin 1 also. Very similar pattern of phenotype with osteomyelitis and fractures, with actually until you get further down, just as with the TREM2, to the Alzheimer's associations and dementias. That's in part because osteoclasts and microglia, osteoclasts that resorb bone, and the microglia in the brain that we know are very important for the development of Alzheimer's are both macrophages. And there's substantial overlap in the genes expressed in osteoclasts and microglia, and in the genetic architecture of their gene regulation. And so when something is going wrong in microglia that will ultimately increase risk of Alzheimer's and dementias, there are sometimes also things going wrong in osteoclasts that increase risks of bone resorption and osteomyelitis. Additional of the genes implicated in Alzheimer's show the pattern of phenome association. And the beauty of a biobank like BioView linked to electronic health records is that while we have these 280,000 people with EHR phenome and genome data, and we can do these studies now in 120,000 of our samples, we've got 2.8 million more people with just the electronic health records, and more than 800,000 of those have what we call a medical home at Vanderbilt University Medical Center with really cleaned data. And that just means that they have at least 3 medical codes in 5 years, so they're getting a lot of medical care, at least at some period of time. And that's a lot of data where we can investigate findings like this, which we have now done. And you can really iterate between what you find here in just the phenome data and ways to validate that in the genetics, or starting with the genetics and looking at validations in the larger data. 0.8 million subjects with the data, and we actually focused much more heavily on the samples with the medical home, the 800,000 with the medical home. We know that the age at diagnosis for osteomyelitis is on average 15 to 20 years earlier than a diagnosis of dementia, and that subjects with osteomyelitis diagnosed before age 60 have a 4 to 6 increased risk of getting a dementia diagnosis after age 70. So this pattern is something that is potentially of larger interest when we think about how we might go about developing drugs for Alzheimer's, because by the time people actually are expressing symptoms of Alzheimer's, it may be too late to reverse consequences. But if we had pleiotropic earlier phenotype that we could follow, we might have a better opportunity to look at drug development in new ways. So, and these are the kinds of studies that you can think about doing in silico first, to see whether there might be any existing drugs that postpone these osteomyelitis diagnoses. The phenome, in fact, is the biggest surprise since I've been here. I've been at Vanderbilt now 5 years and very excited about working in the biobank, but it's the phenome that is amazingly rich. And I'm I just can't believe how detailed these diagnoses get. This is an ICD-10 code for sucked into jet engine, subsequent encounter. Illustrated by a talented artist. And you can see the words here, not again, for the subsequent encounter, being sucked into the jet engine here for a second time. So the phenome is a really fun place to play. And current phenome projects ongoing at Vanderbilt include one that Lisa Bastarash has been doing to really use a lot of expert opinion for the mapping of ICD-9 and 10 codes that have been popularized for phenome-wide association studies, a distillation, if you will, of the tens of thousands of billing codes to a much condensed set of fee codes. So that's getting close to being ready for prime time use. But Wei-Chi Wei is also doing some very cool research, really considering the probability of diagnoses for an individual rather than something discrete as a diagnosis. And I think both of these are really exciting potential future developments for how we will be using phenome in biobank studies when the phenome is coming in from electronic health records. And I want to show you just a quick and dirty project that we've been looking at for certain of the drugs, to look at how the phenotypes match up with what we know about what the drug's supposed to be doing. So if we consider the fee codes by group, we have this sort of color wheel, and it tells you what these different classes, so many different codes are all incorporated within these broader categories. And so 8% of all these fee codes fall into the cancer group, and 9% belong in the cardiovascular group, and so forth. So we're interested in the drugs that are the GLP-1R agonists used to treat diabetes, but that have a favorable profile for cardiovascular. And so there's many ways to look at this. We've been focusing on the fact that there will be pleiotropic effects. And so this is a postdoc, Meg Shuey, who's been doing these studies. And we're just going to take all the genes in this GLP-1R pathway and probe using our genetically predicted expression of genes. We're interested in what medical phenome is associated to the genetically predicted expression of the 40 or so genes in this pathway. And there is some very modest enrichment of significant phenome for GLP-1R pathway genes relative to the baseline proportion of phenome in this category. So for endocrine and diabetes related codes, 6% of all codes fall into that category, and 8% of the significant associations for GLP-1R pathway genes fall into that category. But there's a lot more enrichment of the cardiovascular fee codes. And in fact, when you look at not just GLP-1R, but other members of that pathway, especially those immediately upstream and downstream in their activities related to GLP-1R, it's all cardiovascular phenome. If you looked at the phenome associated to the genetically predicted expression of those genes and had to guess, you'd guess that they were developed to treat cardiovascular disease. And maybe somebody would get the idea of looking at them for how they might affect blood glucose levels, but you'd think that they were primarily driving treatment of cardiovascular disease. And in fact, of course, that's one of the reasons they're such successful drugs. They were among really the first drugs to show a favorable outcome for cardiovascular risk factors in the diabetic subjects who were prescribed these drugs. Another very cool set of research at the phenome end of things was the development of the phenotype risk score, analogous to those of you who do to a genetic risk score. This was a way to look at Mendelian disease phenome in a principled way across a biobank. Ostensibly, you could use it to try to diagnose patients with potential Mendelian conditions that were undiagnosed. And it is just like a polygenic risk score, a summation of a set of phenotypes that have been implicated in disease. I'm getting a message that I'm unstable, and I think I should stop my camera, so I'm going to try to do that. Yeah, okay. That usually helps. Sorry, when all the kids start in with school, I lose bandwidth. But we've now taken this concept of a phenotype risk score, so just these, a summation of what phenotypes an individual has with the weight reflecting initially how rare or common that phenotype was, but could be other things as well, other ways of weighting, and are looking at things that are not quite diseases, but rather syndromes. So VACTERL, for example, requires, a VACTERL diagnosis requires at least 3 of the following congenital anomalies, vertebral defects, etc. And so one of my postdocs has created a phenome risk score for VACTERL and is using that to try to find genetic risk factors for VACTERL based on the phenome risk score. And so you see here with the phenome risk score across individuals in BioView, in the 90,000 samples that we have mega genotype data on. So the green indicates an EHR confirmed case of VACTERL, but the gray dots, gray bars really are subjects without a diagnosis. So you can see that the patients diagnosed tend to have higher Phe codes, and those that have lower Phe codes are actually fairly recently a part of our electronic health record system, or were in the system for a very short period of time. And then this is the density for those without VACTERL. So most of them have very low codes with a tail going off into higher codes, whereas those with VACTERL diagnoses confirmed by chart review, so not just the codes, have a much higher average score, highly significantly increased. And so, so she's doing GWAS and PredictScan, this predicted expression sort of study of VACTERL in this way, but also very explicit tests of some predefined hypotheses on the NADH pathway. And she's conducting similar studies on many other syndromes that should have these very similar patterns of not very many genes having been implicated yet, despite having been extensively studied, with many of the cases coming from de novo mutations in different genes, which, for syndromes where there are multiple congenital anomalies, that wouldn't be surprising at all. And so we think that by using these phenome risk scores in larger scale data, we might be able to then interface with the Mendelian sequencing centers and undiagnosed diseases network to help identify subsets of genes that may be much more likely candidates based on their association to these phenome risk scores in a large scale biobank. And then there's just other really cool things you can do with a biobank. We've been thinking a lot about might be able to move polygenic liability across species to some of our favorite model systems. And so a team effort, we've been working with Gary Churchill at Jackson Labs and Alan Addy and Mark Keller at the University of Wisconsin, who've done a lot of studies in the diversity outcross mice that include a lot of transcriptomics collected very recently on very large samples of islets and brain samples, heart, muscle, adipose, all the tissues that you'd want to look at for something like cardiovascular disease. And then my postdoc Dan Zhao and a statistical geneticist at Vanderbilt, along with Hockey M and Abe Palmer, former colleagues from the University of Chicago. Abe's now at UCSD, who are working in the rat. We're all very interested in doing this. And the idea is that while you can't, the SNPs are not the same in mice or rats and humans, but the genes are almost all the same, in the sense that 85% of the genes have a one-to-one correspondence, mouse and human. And most of the rest of that 15% are, have a one to more than one correspondence in one direction or the other. Very few genes are actually represented only in the mouse or only in human. And so what we're considering is use of a polygene risk score to do this liftover, rather than the polygenic risk score that's SNP based. So a polygenic risk score can be estimated from just the summation of risk alleles by some weight that may come from the GWAS. And you'd have a similar thing for a polygene risk score, where you're summing over the genetically predicted expression of a gene by some weight that would come from a TWAS over the available data for that given phenotype. And this you could really do a liftover cross species for if you had similar prediction models built in the mouse and rat as you do in human. And so Dan Zhao has built these prediction models in the DO mice for the transcriptomes derived from islets. And relative to just random predictions, we get very high quality prediction in mice. In fact, better quality prediction than we get in humans, almost certainly because the transcriptomes are more highly heritable in mice because the environment is much more highly regulated and more similar from mouse to mouse than our exposures are in humans. So the prediction, the transcriptome prediction works very well in mouse, and Hockey's reporting very similar success in the rats, works very well in rats. Then the key question is, can a polygene risk score do anywhere near as well as a polygenic risk score? Because polygene risk score is working only through the transcriptome, whereas polygenic risk scores could be using coding SNPs as well. But it turns out that for all of the islet traits measured in the mice, the polygene risk score works as well or better than the polygenic risk score. And we think that that's in part due to the fact that we do quite a bit of pruning for polygenic risk score construction, such that we're often using a single SNP from a locus that may have many independent signals that are acting to affect risk of disease through the transcriptome. So we're losing some parts of the transcriptomic signal in a PRS, even though we're gaining some aspects of the coding part of the signature. And with the polygene risk score, we're doing much better at the part of the PRS that is really being driven by transcriptomics, even without the coding variation. But for something like BMI, where we only had the islets, we really don't get high quality prediction performance from a polygene risk score. And we think that's because some of the relevant genes are simply not expressed in the islets. And so we're going to be building this out now with the RNA from other tissues to see whether we can do a better job by including more genes from more tissues. Of course, there's still a lot of challenges as we try to do this. In general, our predictions are going to be built in the human data from much, much larger sample sizes. If we want to translate BMI from human to mouse, for example, we should be able to build a much better prediction model in humans because we'll have data from millions. In addition, we do recognize the fact that we have different prediction performance by species for different genes, and we'll have to take that into account. So the weighting must reflect not just the magnitude of the contribution the gene makes to the phenotype of interest, but some aspect of the differential in our ability to predict the gene's expression in the two species. But this idea of multi-gene probes, whether it's a full polygene risk score or just a gene set from a pathway, the TGF-beta signaling pathway, for example, or trying to look at the phenotype for all implicated in congenital heart disease or VACTERL, we think that using these multi-gene probes is also going to be a really interesting thing to look at across the biobank. And from one of our recent papers, where we've identified a bunch of phenome associated to collagen, the failure to traffic collagen genes that strengthen and cross-link extracellular matrix, we're very anxious to build a multi-gene probe to look at how similar the medical phenome might be to what we've seen, the failure to traffic these collagens. Some of the other research involves that you can do in a biobank. You can look back at archaic hominins to understand how the part of our genetics inherited from Neanderthal and other archaic hominins were likely to affect medical. And so this has been really fun research that we have done in collaboration with Tony Capra. I mentioned the study recently published in Nature Medicine, where we really used the phenome from both our biobank and the UK Biobank to enrich our understanding of some phenotypic consequences of knockouts that were first studied in zebrafish, but that turn out to have Mendelian diseases associated with them, where our colleagues studying these Mendelian cases were actually able to go back and do guided reevaluation of the patients to discover that they indeed had many of the same additional phenotypes that we characterized in the biobanks. And that was a really fun study that brought in a lot more biology from the zebrafish end and got to the mechanism of how these genes worked and why we were seeing so much pleiotropic phenome in the biobanks, and then also in the kids with mutations in these same genes. I'd like to close just talking the last few minutes about some community collaboration and creating a unique resource for induced pluripotent stem cells from the biobanking resources. So we approached the community advisory board about whether it might be possible to extend BioView to create induced pluripotent stem cells. And the community board was very concerned with the quite different nature of cell line creation relative to using DNA samples to do genetic studies. But they invited the faculty to address the board. First, the faculty went and described the rationale for why they wanted to be able to create these induced pluripotent stem cells. And when they provided that rationale, the community board suggested that, look, you just admitted you don't even know for sure if it'll work. Why don't you try a pilot and come back to us and we'll talk about it some more. And so they did. And just to elaborate a little bit on the rationale that was described for the cell creation, part of it is to study those rare, unique individuals with large effect mutations that get identified in the context of the biobanking studies, where you want to see those particular individuals. But a second important rationale is to study really the biology associated to polygenic liability to disease. And I'm showing here a histogram of type 2 diabetes polygenic risk score, very normal distribution. And the idea was to really create iPSCs so that we could study across many different cell types important in diseases like diabetes or Alzheimer's. What's different about the people with high polygenic liability, in this case to diabetes, relative to those with low polygenic liability and those in the mid ranges, so throughout the rest of this distribution. So instead of studying a single large effect mutation, we're studying the people, 4 or 5 standard deviations out, because there are people that far out, in polygenic liability, to really learn what effect that has on beta cells, muscle cells, adipose. You can see in this, for the different levels of standard deviation grouping, the blood glucose levels, of course, are going up as the liability increases, going down in controls as risk declines. We see the phenotypic consequences even here, but it would be a really unique opportunity to start to understand polygenic risk at scale by creating these iPSCs. So after the pilot, the faculty provided the results of the pilot studies showing that the iPSCs could indeed be obtained from leftover blood and successfully used to create and differentiate the iPSCs. And they were able to demonstrate some very preliminary things they were learning from this pilot application. And so the board agreed to a trial within Vanderbilt of creation of these iPSCs, with the stipulations that the cells were not to leave Vanderbilt and to be destroyed at the conclusion of the study, mindful of the Henrietta Lacks debacles around cells. And so we're proceeding to try and get these kinds of studies going. I'd like to take questions now and thank everybody for their attention, and I thank my funders also, and the community boards that really enabled BioView to be created and continue to be a partner in our research with the Biobank.
Thank you. This was a phenomenal talk. I will clap for you since we don't have virtual clapping, but I really, really enjoyed this talk. So we have one question that has been submitted. If others have questions, please do that. The comment is, wonderful talk, Nancy. Thanks so much for presenting today. I found the Alzheimer's osteomyelitis example particularly intriguing. Do you know of any companies looking at the implications? And have you looked at parallel associations between osteomyelitis and other dementias? And I will take that a step further. As someone who does schizophrenia research, which has hallucinations as one of the hallmark symptoms, have you taken it beyond dementia to other psychiatric illnesses?
So because of the correlations in the phenome, you definitely, I mean, we don't anything associated to Alzheimer's or in fact any dementia that is not associated to all of them. So some of the Alzheimer's disease aficionados say that's because that's all contamination. But I think even when they've tried to use findings that are very clear from autopsy confirmed Alzheimer's patients, those genetic risk factors are genetic risk factors for vascular dementias as well, for other forms of late onset dementias. So it's a little bit hard to know. And the fact is that within EHRs, you see a whole cluster of diagnoses that have high correlation to the genetic architecture associated with Alzheimer's disease. So I would be astonished if you didn't see some degree of hallucination associated. I can say we see that whole symptomatology within this 4 to 6 fold excess risk. But I think that's just the nature of the strong phenotype correlations for diagnoses. People are sometimes getting the hallucination diagnosis before they get the later dementia diagnosis. And therefore you're seeing these phenotype correlations in that way. I do, so this research is ongoing, and among the things that are being really used, the full EHR to look at drugs that people take for other things, at whether there's any support that any of those drugs might delay onset of either the osteomyelitis diagnosis or the Alzheimer's diagnoses. I always report unpublished research. Well, although we're very close to submitting something around some of these findings. And so I don't really know whether any companies might be working on it or not. I always love to see others pick up and look at some of the things that I think are worth looking at. So I wouldn't be sad if other people started looking at it.
Yeah, hopefully we have someone from Michigan Drug Discovery that is on the webinar today that can kind of take that torch and carry it even further. There is another follow-up question about Parkinson's. So we talked about, other psychiatric illnesses. Parkinson's a little bit different, but definitely, very much involved in the, the mechanisms behind those. So.
Yeah, so there does appear to be some degree of shared genetic architecture among some of the dementias, some of the other neurodegenerative conditions like Parkinson's, but very clearly each has some of its own genetic architecture.
Right.
And I think it's going to be really interesting to see whether the parts that are shared reflect some of the what you might call resilience to later onset brain disease that might be shared across different neurodegenerative and dementia phenotypes. People are looking at things from both directions, as it were, the sort of unique and shared genetic architectures increasing risk of disease, but there's almost certainly then immune components to a number of the neurodegenerative conditions and dementias. And this concept of resilience that may in part relate to the ability of the immune system to respond to the lesions effectively, but may involve other things as well. So I think all of just fascinating research.
Have you done, being a pharmacist, I'm very interested in medications, but have you ever done any kind of longitudinal analyses that would allow you to maybe look at potential pathways of how our medications work? Because I think probably what we know about how they work is pales in comparisons to how they actually really work.
We've been talking about trying to push out sort of like a pharmacoome cross to the phenome to take the genes, to take the drugs that have purported gene targets and really pull that together in a way that people can query. I should have updated my slides to have we are making public a version of the portal with this gene by medical phenome catalog, and but packaging up the part of it that's relevant to the gene targets of drugs, I think would be really interesting. Megan is actually a pharmacologist, expert in pharmacogenetics, and is very interested in doing that. So I think she'll have it done before long.
Yeah, plus you have Dan Roden there, so.
Yeah, yeah. Well, and so one of the things that people are starting to do in follow up to GWAS and the TWAS or PredictScan kinds of analyses are to really look at whether any of the top genes implicated are drug targets, and if they are, whether those drugs seem to have the expected effects based on the directions that seem associated with the disease. And so I think recent scale GWAS hypertension diabetes had those studies based on our biobank as part of the outcomes. I'm losing my connection again.
Sorry, that's fine. So another question about what's BioView's policy of sharing data with outside investigators?
So we're generally allowed to share freely with academic investigators, and Vanderbilt does a lot of collaborations with industry, and those can be at the level of the investigator or with a company that was developed to commercialize BioView to make it easier for pharma and biotech to be able to interact with the biobank. So yes, data can be shared, and certainly with collaborators, we've shared, all kinds of summary information as well as data at the genotype level when it's necessary.
And then one last question in our last couple minutes about if you have plans to collect expansive survey data like the UK Biobank questionnaires, and what might be some of the challenges.
Yeah, no, so we do have My Health at Vanderbilt, and there are a lot of people who would love to collect more data in that way. With some of the young faculty that I work with, one is doing these very cool studies in rhythm and music as the precursors to communication, relationship between the genetic components to rhythm and music and speech and language disorders. She's got these very cool tests that can be done by computer that measure rhythm and musical abilities, and it would be so fun to deploy those through My Health at Vanderbilt and be able to link that up to all the genetic data. And it's just, it's a bandwidth issue for the people who work in that space as to, what things get deployed there first, and it's going to be wearables first. I think there is a lot of interest in being able to allow people, if they have the interest and ability, to interface with that portal and do surveys, do these kinds of fun tests that relate to rhythm and music and things like that too.
Great, thank you. Wonderful talk again. Thank you so much.
My pleasure. I'm looking forward to the day.